I'm very happy and honored to be here for this lecture. I'm the Dr. Shuai Tan from the Department of Hematology, Xuanwu Hospital, Capital Medical University, Beijing, China. Today we'll talk about the new immune target derived from the platelet, PF4, and the TGF-beta. 3 parts. Let's go to the first part, the background. We talk about platelet. Platelets release from macrocytes in the bone marrow. The smallest blood cells with the allele structure, and it is easier for activation from the domain to the activation. The platelet play a role in maintaining hematosis and forming thrombosis. And we go to the T cells, the CD4 T cells, the CD8 T cells. T cells can be activation and degradation for the first signal and the third signal. T cells are a major driving force in immune reaction and inflammation. For CD4 T cells, in which Th1 Treg cells are pro and anti-immune reaction inflammation respectively. Now we notice most of the active T cells produce Th1 cytokines interferon gamma, which active macrophages and vascular T cells. Tregs modulate the process, and the T cells at the caprate site. So we know the platelets participated in all stages of forming thrombosis. All stages, the 4 stages, the platelets for the hematogenous thrombosis, the T cells immune reaction and inflammation. So we recognized the platelets and T cells as partners on the immune response and inflammation. When we retrospect the brief history of the antiplatelet, we divide into the 4 parts: the antiplatelet prehistory area, the monotherapy antiplatelet area, and now the dual therapy antiplatelet area. So in the 2025, what is this time the area? So we go to the second part, platelet T cell crosstalk. Now we know the more and more researchers and scientists focus on this field. We know the interaction between the platelet and the immune response of passages. The platelets participate in immune responses with immune cells. The platelets and T cells aggregates. Now in Kalinska, we built some models. We know the platelets exert distinct regulation on effector cells response of CD4 T cells, enhancing T cell immune response in healthy individuals by release different types of the suitable media, such as PF4, TGF-beta, etc. These mediators have promoting or inhibiting effects on different kinds of T cells. So the platelet as an enable coordinator for immunology. So in time, we have the 3 models of T cells. No matter the line model, the circular model, or the asymmetric division model, there are 3 important cells: the T-naive cell, central memory cell, and effector memory cell. So if you want to know the behind mechanism behind the crosstalk effect of platelet T cells, we should research on the TEM, TCM, and T-naive. And in fact, we spent a long time, almost 5 years. We found the TEM first discovery of platelet release PF4 and sex3 binding to initiate AKT P7T5 mitochondrial pathway regulation of the TEM. The PF4 activates the mitochondrial metabolism. For the TCM crosstalk, we discovery of the platelet at TCM with delayed cell proliferation and immune response. And for the naive, it's much more complex. It's all discovery of dual regulation. It's also not only by the PF4, also by the PF4 and the TGF-beta. There's a dual regulation here. So the brief summary of the mechanism is 3 points. 1, the CD4 TEM, TCM, T-naive. The mechanism of them are not totally the same. The mitochondrial pathway is very important and depends on the PF4 only, like the PF4 or the dual regulation PF4 and TGF-beta. It depends or based on the PF4 concentration. And in last year, in 2024, the Professor Yamato from Japan published a very outstanding paper in Science, PF4-induced Th1 Treg polarization suppressed anti-tumor immunity is also based on the PF4 theory. So if you have time, I recommend you struggling to read this paper. It's very interesting. So we focus on the plastic anemia. Idiopathic typemia and bone marrow failure. And now the research is focused on the CD8 T cells about the interferon gamma. But how it attacks the bone marrow or what kind of the mechanism is not clearly. So the treatment is IST, but the challenge is still here. Slow onset of action, significant side effects, long treatment duration, and approximately 1/3 of patients unable to achieve long-time survival. So we need to find a new target or a much more precise target for the disease. So based on the platelet PF4 T cell crosstalk mechanism, and combined with the fact that patients with AA may experience immune overreaction, and we conducted a survey of global literature and research and the first proposal, the hypothesis of platelet T cell and bone marrow failure. This is the figure we hypothesis. So the platelet from the dormant to active release a lot of different kinds of proteins, PF4 TGF-β, rings, and etc. And can also bind with the T cells like CD4 T cells, maybe CS3 or CS5, but CS5 not advanced or much more expression all the T cells, only in several of them, like the TCM. They may active the active PCL alpha TFAM, the signal pathway, and for the CD8 T cell, the PF4 also can bind the S3, can also active the signal pathway, and then did their function or differentiation, like the Th cells or Treg cells. For Treg, it may suppress because it can protect the bone marrow. So then, overall, it can release or influence in cytokine interferon gamma, and then to attack the HSC or the bone marrow failure. So we want to confirming potential new signal pathways in bone marrow failure, and we also do some research, investigate this potential new regulated mechanism. Now I want to introduce some of them. So we built the immune-mediated SAA mouse model, two groups. One is the experiment group, SAA, one is control group, next TBI. And after 12 or 13 days post lymphocyte infusion, the BMF model will be built. And based on this model, we did single cell sequencing or cross-analysis of single cell data with humans. And flow cytometry, Western blot, and also based on the human database, we also did some meta-analysis. We go to the first part, BMF mouse model, single cell sequencing. This is also first application of this kind of this method in neural BF4 models to comprehensively map the immune landscape, including many different kinds of cells. This is for the 4 group, the TBI group, BF4 group, wild type, and aged, because we also want to observe the difference between the older aged people and the AA patients. So for the T cell and the macrophages, we notice in the BMF group, the CD8 stress or CD8 effect are much higher here, and for the aged, the Treg are much here. So for Z3 and Z5 expression, we notice it expressed in the BMF group. For the macrophages, we did the dimensionality reduction. We found one group here in the aged group is very, very high, but it is not the macrophage type 1 or macrophage type 2. So we notice it high expressed the CD68, PF4, S100A8, APOL1, CCI3. It may be a new kind of the macrophages. This X3 expression, CD4, CD8 on the BM4 group. And this is the CD68, PF4, C1QA, S100A8. Here is a new kind of the macrophages maybe. And we also did the partial time analysis. We found in the PM4 group, we found the derived was decreased, and the CD4 and CD8 T cells are increased. And for the macrophages type 1 were much high in the experiment group, but aged, we find much about the new kind of the macrophages. So the CCI, the cell-cell interaction for the major types, we find the TBI, the numbers are much high, but the BM4, the intensity and much stronger. This is the details of the CCI in different groups. And then for the platelet, we also found in the BM4 group, that is much high. So it is shows the platelets also did an important role in this process. So for the highlight, we also found this is specific retention analysis of platelets and the mitochondria. We also proposal of AA immune imbalance seesaw mechanism between the PF4 mediated platelet and the T cells and NK cells or the macrophages crosstalk theory. So we also did some research for the human data. We found 2 outstanding papers on the PubMed. So we also downloaded their data for the cross analysis. The first paper, we found a total of 10 cell types were identified, similar cell types were identified in both disease and health group. And compared with the health group, the proportion of the neutral 1 decreased and the proportion of neutral 2 increased in the disease group. And compared with the disease group, the MD2 was absent in the health, and the proportion of the MD1 was much high in the disease group. And for the gene expression, the neutral 1 T cell gene expression change was the most significant. The differential analysis transcription changes in MD2 occurred during the disease process. We also did some functional enrichment analysis. The MD1 differentiated into the dendritic cells, MD2 into the monocytes. So the expression panels of macrophages marker genes in MD cells, we found the M1 subtype were much high expressed. It is matched with our animal single cell sequencing. The M2 showed little to no expression. The procedure time analysis during the disease, MD cells undergo reprogramming from the MD1 to MD2. And for the TCM8, CD8 also find much high in the disease group. It's also matched with our animal study. SF3, SF5 are also much more expressed. The functional enrichment analysis here. For the second paper, we also did the cross analysis, but for the lecture time limitation, I will skip this very fast because I want to show you some of our IF and the flight cytometry result. Let me skip this first. Before the IF result, I want to show the time turning point. We detect the hemoglobin, platelet, white blood cell counting. We found in the day 6, this time turning point. So we did the IF in the day 6. We detect the CD4, CD8, M1, M2, or some signal pathways proteins. We go to the lymph node. This mitochondrion represents the red. The green represents CD8. We can obviously find the CD8 mitochondria is much high than the control group. This also similar result in the spleen. This is a bone marrow. And for the CD4, the red is also represent the mitochondrion, the green for CD4. When we combine, apply them, we find the CD4 and the mitochondria are much high. But which kind of the T cells, the Th1 or Treg? We need to do more research about on it. So this is the spleen. And for the CD4 T cells, the mitochondria, we found this is much high in the experiment group. And this is the flow cytometry result. We also did the experiment on these many markers. I also want to show some of them. For the CD4 T cell, TCM, TEM, T naive, we also find CD4 TCM are much high. And SEX3, the expression, are also much high in the TCM, but SEX3-5 not so much high. So maybe this is not the same. And for CD8, also the naive, the TEM, TCM, T naive, the TCM also much high in the AA group compared to the TBI group. And if you can remember before, it's in the human data, the single cell data is also matched with here. So we also did some Western blot. It's better active in the different groups. We also do some proteins like the P53, or this is also others proteins, signal pathway proteins. The overall standing also showed in the SAA group, there's almost no atherosclerosis or some lipid area. So we also doing the much more IF and WB experiment. For the clinical research, we found the different we also find different when we use the TPY plus IST in patients with AA. The immunology function not only for the AA, also for the another diseases. So we built an experiment group, PF4 positive TGF-β1 flux mouse. We found the platelet specific TGF-β1 deficiency aggravates the chlorosis, vascular inflammation, and hypocholesterolemia in mouse. And in this group, we will found the platelet specific TGF-β1 deficiency and aggravates the chlorosis. This is overall standing. So in the last part, the take-home message. So we will remember the 3 parts or 4 parts. The first is platelet, are key factors, not only the hematosis and thrombosis and the immunology cells, T cells, macrophages, neutrophils, and etc. And the PF4 TGF-β range or others, the mediators from the platelet. The signal pathway may be the mitochondrial energy metabolism. In the sclerosis, we will find the platelet derived TGF-β is prominently atheroprotective. So in summary, this is currently one of the strong hypotheses for present regulation of the immune cells. Also want to introduce our clinical research part, leading by Dr. Wanning Sun, is our director at PI. And we can do a lot of things for the patients, the immunotherapy, including CAR-T and HSCT, etc. And we also are looking forward for the academic exchanges and collaboration. Call for papers, one of the good journal, BND, and for the JOVE, we will have the hematology collection. If you have some good papers, please contact me. This is the introduction for the brain network disorders, BND. So, thank you very much.